Welcome to Sleepy Science Hub. I'm delighted you've found your way here tonight, to this peaceful corner where nature's most fascinating mysteries meet the gentle art of restful learning. As you settle into your most comfortable position, wherever you might be in the world right now, take a moment to let me know in the comments where you are and what time it is as you listen. There's something wonderfully grounding about knowing we're sharing this quiet moment of discovery together, connected by our curiosity about the remarkable world that surrounds us. Tonight, we're going to venture into one of nature's most patient and elegant hunting grounds, the world of carnivorous plants. But don't let the word carnivorous concern you. We're not exploring anything frightening or violent. Instead, we'll discover how these remarkable plants have evolved some of nature's most gentle and sophisticated trapping mechanisms, operating with the quiet patience of master gardeners who understand that the best things in life come to those who wait. Now, get comfortable. Adjust your pillows, pull your blankets close, and let your breathing settle into its natural, peaceful rhythm. Take a slow, deep breath. Feel your body releasing the tensions of the day. And let's begin this tranquil journey into the hidden world of nature's most patient hunters. As you lie there in comfort, your breathing gradually slowing and deepening like the gentle rhythm of morning mist settling over a quiet bog. Imagine yourself walking along a peaceful woodland path in the early hours of dawn. The air is cool and fresh, carrying the soft fragrance of moss and damp earth, and your footsteps make barely a whisper on the carpet of fallen leaves beneath your feet. You're approaching a very special place, a hidden garden where some of nature's most remarkable plants have learned to supplement their diet in ways that seem almost magical in their elegant simplicity. This is not a garden of towering trees or showy flowers, but rather a humble wetland where the soil is poor in nutrients and the competition for resources has led to some of the most creative adaptations in the plant kingdom. Here, in the acidic waters of bogs and marshes, along the edges of quiet streams, and in the sandy soils of coastal plains, carnivorous plants have discovered how to turn the tables on the animal kingdom in the most gentle and patient ways imaginable. The morning light filters through the canopy above, creating a soft, dappled pattern that dances gently across the landscape as a barely perceptible breeze stirs the leaves. In this peaceful setting, you begin to notice plants that seem somehow different from their neighbors. Their leaves shaped in unusual ways, their colors perhaps a bit more vibrant, their overall appearance suggesting adaptations that serve purposes beyond simple photosynthesis. These are the carnivorous plants, and they represent one of evolution's most elegant solutions to the challenge of surviving in nutrient-poor environments. As your eyes adjust to the subtle details of this quiet ecosystem, you begin to understand that carnivorous plants are not aggressive predators in any conventional sense. They don't chase their prey or actively hunt in the way we might imagine predators behaving. Instead, they have mastered the art of patient invitation, creating beautiful and ingenious traps that work entirely through passive mechanisms, structures so perfectly designed that insects and other small creatures are naturally drawn to them, often finding themselves gently captured before they even realize what has happened. The evolutionary story of carnivorous plants is one of remarkable ingenuity arising from necessity. In environments where the soil lacks essential nutrients like nitrogen and phosphorus, nutrients that most plants obtain through their root systems, some plant species began to experiment with alternative feeding strategies. Over millions of years, through countless generations of gradual modification, ordinary plant structures like leaves began to transform into sophisticated trapping mechanisms that could supplement the plant's nutritional needs by capturing and digesting small animals. This transformation didn't happen overnight, and it didn't involve any sudden dramatic changes. Instead, it represents the beautiful patience of evolutionary processes, where small advantages accumulate over vast periods of time until structures that began as simple adaptations become marvels of biological engineering. A leaf that was slightly more curved than its neighbors might have occasionally trapped rainwater and the occasional insect. Over countless generations, Those plants with slightly better water-trapping leaves had small nutritional advantages that allowed them to reproduce more successfully, gradually leading to increasingly sophisticated trapping mechanisms. The beauty of carnivorous plants lies not just in their unusual feeding habits, but in the incredible diversity of solutions they have evolved to solve the same basic problem. From the elegant pitcher-shaped leaves that create drowning pools, to the snap traps that close with remarkable precision, to the adhesive surfaces that hold prey gently but firmly, carnivorous plants demonstrate nature's creativity in finding multiple pathways to the same destination. Each type of trap represents a different approach to the challenge of supplementing nutrition, and each works with its own particular form of patient effectiveness. As you continue to explore this gentle garden in your mind, your breathing now completely relaxed and natural, you begin to appreciate that carnivorous plants have not abandoned photosynthesis, the process by which most plants convert sunlight into energy. Instead, they continue to photosynthesize just like their non-carnivorous relatives, using the energy from sunlight to power their growth and basic metabolic processes. The insects and other small creatures they capture serve as supplements to their diet, providing the additional nutrients that are scarce in their challenging environments. This dual feeding strategy, combining photosynthesis with carnivory, makes these plants particularly fascinating from a biological perspective. They represent organisms that have successfully bridged the gap between the plant and animal kingdoms in terms of nutritional strategy, while maintaining all the essential characteristics that define them as plants. They remain rooted in place. They continue to produce their own energy through photosynthesis, and they reproduce through flowers and seeds just like other flowering plants. The habitats where carnivorous plants thrive are often places of particular beauty and tranquility. Sphagnum bogs, with their carpet of soft moss and mirror-like pools of tea-colored water, create environments that seem almost otherworldly in their peaceful isolation. These bogs develop over thousands of years as organic matter accumulates in poorly drained areas, creating acidic conditions that slow decomposition and result in nutrient-poor soils that favor carnivorous plants over their more conventional competitors. The chemistry of these bog environments is remarkably stable and predictable, with pH levels typically ranging from 3.5 to 4.5, quite acidic by most standards, but perfect for the specialized plants that call these places home. The water in these environments is often naturally soft and low in dissolved minerals, creating conditions that would be challenging for most plants, but ideal for species that have evolved to capture their nutrients from animal sources rather than from the soil. In these peaceful wetland gardens, time seems to move at a different pace. The seasonal changes occur gradually and predictably, with each stage of the year bringing its own particular opportunities and challenges for the carnivorous plants that live there. Spring brings warming temperatures and longer days that trigger new growth and the formation of fresh trap structures. Summer provides abundant insect activity that represents the peak feeding season for most carnivorous plants. Autumn brings the gradual preparation for winter dormancy, while winter offers a period of rest and energy conservation that allows these remarkable plants to survive the cold months and prepare for another growing season. The insects and other small creatures that become prey for carnivorous plants are typically drawn to the traps by a combination of visual, chemical, and tactile attractions that work together to create irresistible invitations. Many carnivorous plants produce nectar or other sweet substances that serve as legitimate food sources for visiting insects, creating a complex relationship where the plant provides real benefits to many of its visitors while capturing only a small percentage of them. This relationship represents a delicate balance that has evolved over millions of years, ensuring that insect populations remain healthy and abundant while providing carnivorous plants with the supplemental nutrition they need to thrive. The process of digestion in carnivorous plants is remarkably gentle and efficient, involving enzymes that break down captured prey slowly and completely, extracting every possible nutrient while leaving behind only the most indigestible components. This process can take days or weeks to complete, reflecting the patient nature of these remarkable plants and their ability to extract maximum benefit from every capture. The digestion process itself is not violent or disturbing, but rather represents a natural recycling of nutrients that benefits the entire ecosystem. The study of carnivorous plants has provided scientists with invaluable insights into evolutionary biology, plant physiology, and ecological relationships. These plants demonstrate how evolutionary pressures can lead to remarkable innovations, how organisms can adapt to challenging environments through creative solutions, and how predator-prey relationships can evolve to be surprisingly gentle and mutually beneficial in their broader ecological context. Many carnivorous plants produce beautiful flowers that seem to contradict their predatory reputation, creating displays of color and fragrance that rival those of any ornamental garden plant. These flowers serve the same reproductive functions as flowers in other plants, attracting pollinators that help ensure the continuation of the species. Interestingly, the flowers are typically held well above the trap structures, ensuring that beneficial pollinators are not accidentally captured by the very plants they are trying to help reproduce. The conservation of carnivorous plant habitats has become increasingly important as development pressure and environmental changes threaten the specialized ecosystems where these remarkable plants thrive. Many species of carnivorous plants are now rare or endangered, not because they are inherently fragile, but because the very specific environmental conditions they require are becoming increasingly uncommon. Protecting bog ecosystems, maintaining natural water levels, and preserving the delicate chemical balances that these environments require has become crucial for ensuring that future generations will be able to experience the wonder of these remarkable plants. As you continue to rest peacefully in this imaginary garden, surrounded by the gentle sounds of water lapping softly against moss-covered banks and the quiet hum of insects going about their daily activities, you can begin to appreciate the profound patience that characterizes carnivorous plants. These are organisms that have learned to wait, sometimes for days or weeks, for the right opportunity to capture prey. They don't rush or force outcomes, but simply maintain their traps in perfect condition and allow natural processes to bring them the nutrition they need. This patience extends to every aspect of their biology, from the slow process of trap development in spring to the gradual digestion of captured prey to the methodical preparation for winter dormancy. Carnivorous plants operate on natural timescales that remind us of the value of patience, persistence, and working in harmony with environmental conditions rather than fighting against them. In a world that often seems to value speed and aggression, these remarkable plants offer a different model, one based on gentle efficiency, patient observation, and the quiet confidence that comes from being perfectly adapted to one's environment. The relationship between carnivorous plants and their prey represents one of nature's most sophisticated examples of ecological balance. While these plants do capture and digest insects and other small creatures, they do so in ways that support rather than threaten the overall health of their ecosystems. The insects they capture represent only a tiny fraction of the total insect population, and the nutrients released through their digestion processes ultimately benefit the entire plant community through natural recycling processes. As your breathing continues to slow and deepen, matching the patient rhythms of the bog environment around you, you can sense the deep tranquility that characterizes these special places. Here, away from the noise and rush of human activity, carnivorous plants continue their ancient practices of patient hunting, efficient digestion, and careful preparation for seasonal changes. They represent living examples of how organisms can thrive through cooperation with natural processes rather than fighting against them, and how some of nature's most remarkable adaptations can arise from the simple need to find creative solutions to environmental challenges. As you settle even deeper into peaceful observation, your body now completely relaxed and your mind drifting like morning mist across the surface of a tranquil bog, let your awareness turn to one of nature's most profound lessons, the art of stillness. In a world where movement often seems synonymous with life and activity with success, carnivorous plants have discovered something remarkable: that perfect stillness, combined with infinite patience, can be one of the most effective strategies for survival. These plants have mastered what ancient philosophers and meditation teachers have long understood, that there is tremendous power in being completely present, perfectly prepared, and utterly still. Imagine yourself sitting quietly beside a small pool in this bog garden, so still that you've become part of the landscape itself. The water before you is mirror smooth, reflecting the sky with such perfect clarity that it's difficult to tell where reality ends and reflection begins. This is the kind of stillness that carnivorous plants embody. Not the stillness of lifelessness, but the dynamic stillness of perfect readiness, like a master archer who has drawn the bow and now waits for the precisely perfect moment to release the arrow. The evolution of carnivorous plants represents one of biology's most elegant demonstrations of how constraints can spark creativity. In the nutrient-poor environments where these plants thrive, the usual plant strategy of extending roots to gather nutrients from the soil simply doesn't work effectively. The acidic, waterlogged soils of bogs and similar habitats contain very little of the nitrogen, phosphorus, and other essential nutrients that plants need to build proteins, nucleic acids, and other vital molecules. Faced with this challenge, some plant lineages began to explore alternative approaches to nutrition, eventually developing the remarkable ability to supplement their diet by capturing and digesting animal prey. This evolutionary innovation didn't happen quickly or easily. The transition from traditional plant nutrition to carnivory required fundamental changes in plant architecture, biochemistry, and physiology that developed over millions of years through countless generations of small improvements. The plants that eventually became carnivorous didn't suddenly develop complex trapping mechanisms overnight. Instead, they began with simple modifications, perhaps leaves that were slightly more curved and better at collecting rainwater, or surfaces that were marginally stickier and occasionally trapped small insects by accident. As you breathe slowly and peacefully, your awareness expanding to encompass the patient rhythms of evolutionary time, you can appreciate how these small initial advantages gradually accumulated into increasingly sophisticated adaptations. A plant whose leaves could occasionally capture a small insect would have access to nutrients that its competitors lacked, giving it a slight reproductive advantage. Over many generations, this advantage would become more pronounced as the trapping structures became more efficient and the digestive processes more effective. The biochemical innovations required for carnivory were perhaps even more remarkable than the physical adaptations. Plants had to develop entirely new metabolic pathways for producing digestive enzymes, mechanisms for absorbing nutrients from animal tissue, and systems for regulating the digestion process to prevent the plants from harming themselves with their own digestive secretions. These biochemical changes represent some of the most sophisticated molecular engineering in the natural world, comparable to the innovations that allowed some bacteria to develop photosynthesis or some animals to develop complex nervous systems. The diversity of carnivorous plant families suggests that the transition to carnivory has occurred independently multiple times throughout evolutionary history. Rather than representing a single evolutionary innovation that spread to different plant groups, carnivory appears to have evolved separately in at least 5 different plant families, each developing its own unique approach to trapping and digesting prey. This parallel evolution demonstrates that carnivory represents a particularly effective solution to the challenge of surviving in nutrient-poor environments, successful enough that natural selection has discovered it repeatedly. The environmental conditions that favor carnivorous plants are quite specific and create what ecologists call ecological niches, particular combinations of physical and chemical conditions that favor certain types of organisms over others. Bogs, fens, and similar wetland environments provide the perfect combination of high acidity, low nutrient availability, and abundant water that carnivorous plants require. These environments are created by specific geological and hydrological conditions that develop over hundreds or thousands of years, making each bog ecosystem a unique and irreplaceable natural laboratory. The acidity of bog environments serves multiple functions in supporting carnivorous plant communities. The low pH inhibits the bacterial decomposition that would normally break down organic matter and release nutrients into the soil, creating the nutrient-poor conditions that give carnivorous plants their competitive advantage. At the same time, the acidity helps preserve the complex organic compounds that accumulate over time to form peat, creating the distinctive substrate that many carnivorous plants call home. The water chemistry of bog environments is equally important, typically characterized by very low concentrations of dissolved minerals and nutrients. This soft, mineral-poor water creates conditions that would be challenging for most plants, but ideal for species that have evolved to obtain their nutrients from animal sources rather than from dissolved minerals in the soil. The consistency of this water chemistry over time provides the stable environmental conditions that allow carnivorous plant communities to develop and persist. Temperature patterns in bog environments also play a crucial role in shaping carnivorous plant ecology. The thermal mass of water in bog environments creates relatively stable temperature conditions that change gradually with the seasons, avoiding the extreme temperature fluctuations that might disrupt the delicate physiological processes involved in carnivory. The cool temperatures typical of many bog environments also slow metabolic processes, allowing carnivorous plants to make the most efficient use of their captured nutrients. As your awareness drifts peacefully through these ecological relationships, your breathing now is steady and natural as the seasonal cycles that govern bog ecosystems. You begin to understand how carnivorous plants have learned to work with rather than against their environmental constraints. Instead of trying to overcome the nutrient-poor conditions of their habitats, they have embraced these conditions and turn them into competitive advantages through their unique feeding strategies. The light conditions in bog environments often favor carnivorous plants as well. Many bogs are relatively open environments with few tall trees to block sunlight, providing the bright light conditions that carnivorous plants need for photosynthesis, while also creating the warm, sunny conditions that encourage insect activity. This combination of abundant light for photosynthesis and abundant insect prey for supplemental nutrition creates ideal conditions for plants that rely on both energy sources. The seasonal patterns of insect activity in bog environments are perfectly synchronized with the growing cycles of carnivorous plants. Spring warming triggers both the emergence of new plant growth and the beginning of insect activity, ensuring that fresh traps are ready when prey becomes available. Summer provides peak insect activity that coincides with the period of most active plant growth, while autumn brings the gradual decline in insect activity that corresponds with the plant's preparation for winter dormancy. The soil chemistry of bog environments reveals the remarkable specificity of the conditions that carnivorous plants require. The accumulation of partially decomposed organic matter creates soils that are not only acidic and nutrient-poor, but also have very specific physical properties that affect water retention, root penetration, and gas exchange. These soils are typically saturated with water throughout much of the year, creating anaerobic conditions that further inhibit decomposition and maintain the low nutrient conditions that favor carnivorous plants. The mycorrhizal relationships that many plants use to enhance nutrient uptake are typically absent or poorly developed in carnivorous plants, reflecting their reduced dependence on root-based nutrition. Instead of investing energy in complex root systems and fungal partnerships, carnivorous plants have redirected their resources toward developing and maintaining their specialized trapping and digestive structures. This represents a fundamental shift in how these plants allocate their energy and resources compared to their non-carnivorous relatives. The water relations of carnivorous plants reflect their adaptation to consistently moist environments. Many species have relatively thin cuticles and limited capacity for water conservation, adaptations that would be disadvantageous in dry environments, but perfectly suited to the consistently humid conditions of bog ecosystems. This physiological specialization helps explain why carnivorous plants are typically restricted to wetland environments and why they often struggle to survive in cultivation unless their specific water requirements are carefully maintained. The reproductive strategies of carnivorous plants often reflect the stable, predictable nature of their bog environments. Many species are perennial, investing energy in long-term survival rather than rapid reproduction. And their flowering and seed production cycles are carefully timed to take advantage of seasonal peaks in pollinator activity. The flowers of carnivorous plants are typically held well above their trapping structures, ensuring that beneficial pollinators are not accidentally captured by the plants they're trying to help reproduce. The dispersal mechanisms of carnivorous plants are often adapted to the wetland environments where they grow. Many species produce small, light seeds that can be dispersed by wind or water to other suitable bog environments, while others rely on vegetative reproduction through runners, bulbs, or other structures that allow them to spread within established bog communities. These dispersal strategies reflect the patchy distribution of suitable carnivorous plant habitats and the importance of being able to colonize new sites when they become available. The conservation biology of carnivorous plants is closely tied to the preservation of their specialized habitats. Because these plants require very specific environmental conditions that are created by complex geological and hydrological processes, they are particularly vulnerable to habitat destruction and environmental changes. Many bog ecosystems have been drained, filled, or otherwise modified for human use, leading to significant declines in carnivorous plant populations and the extinction of some species from large portions of their former ranges. Climate change presents additional challenges for carnivorous plant conservation, as changing precipitation patterns, temperature regimes, and seasonal timing can disrupt the delicate environmental conditions that these plants require. The specialized nature of bog ecosystems means that they may be particularly sensitive to environmental changes, and the restricted distributions of many carnivorous plant species leave them with limited options for adapting to changing conditions. The study of carnivorous plants has contributed significantly to our understanding of plant evolution, ecology, and physiology. These remarkable plants provide natural experiments in evolutionary innovation, ecological specialization, and physiological adaptation that continue to yield new insights into how life adapts to challenging environments. The biochemical pathways involved in carnivory, the ecological relationships that maintain bog ecosystems, and the evolutionary processes that created such diverse trapping mechanisms all provide valuable lessons for understanding broader biological principles. The cultural significance of carnivorous plants extends far beyond their scientific importance. These remarkable plants have captured human imagination for centuries, inspiring folklore, literature, and scientific investigation. Their ability to blur the traditional boundaries between plants and animals challenges our understanding of natural categories and reminds us that nature is far more creative and flexible than our simple classifications might suggest. As you continue to rest peacefully in this imaginary bog garden, your consciousness now fully integrated with the patient rhythms of carnivorous plant life, you can appreciate the profound lesson these plants offer about the power of specialization and adaptation. Rather than trying to compete directly with other plants in conventional ways, carnivorous plants have found their own unique niche where their specialized adaptations give them decisive advantages. They demonstrate that success in nature often comes not from being stronger or faster than competitors, but from being perfectly adapted to specific environmental conditions and opportunities. The stillness of carnivorous plants is not passive, but actively maintained through constant physiological processes that keep their traps in perfect condition, their digestive systems ready for action, and their overall health optimized for their challenging environment. This active stillness requires as much energy and attention as more obvious forms of activity, reminding us that true mastery often appears effortless, but is actually the result of perfect preparation and constant readiness. The art of stillness that carnivorous plants have mastered extends beyond their hunting strategies to encompass their entire approach to life. They have learned to work with natural cycles rather than fighting against them, to be patient rather than hasty, and to find opportunity in apparent limitation. These lessons apply far beyond the realm of plant biology, offering insights into how any organism, including humans, might approach the challenges of living in harmony with environmental constraints while still achieving remarkable success. As you continue to rest peacefully, Your breathing now flowing with the gentle rhythm of water slowly filling and emptying in nature's most patient vessels. Let your awareness drift toward one of the most elegant and sophisticated hunting strategies in the plant kingdom, the pitcher trap. These remarkable structures represent nature's mastery of fluid dynamics, chemistry, and behavioral psychology, all combined into living chalices that invite, welcome, and gently embrace their visitors in ways that transform what might seem like predation into something that resembles hospitality taken to its logical conclusion. Picture yourself kneeling beside a cluster of pitcher plants, their graceful, elongated leaves rising from the bog like elegant urns crafted by a master potter. Each pitcher is a marvel of biological engineering, its smooth walls curving upward to form a hollow vessel that can hold several ounces of rainwater, secreted fluids, and eventually, the small creatures that will supplement the plant's nutrition. The opening of each pitcher is crowned with a hood-like lid that provides partial protection from excessive rainfall, while creating an overhang that guides flying insects toward the trap's interior. The morning light filtering through the bog creates jewel-like reflections on the surface of the liquid within each pitcher, and you can see that this is no ordinary rainwater. The plant has transformed this collected moisture into a complex chemical solution that serves multiple purposes: part swimming pool, part digestive chamber, part carefully balanced ecosystem in miniature. The liquid appears perfectly clear and innocent, its surface tension creating a mirror-like quality that reflects the sky above and creates an almost hypnotic visual appeal that draws the eye downward into the depths of the trap. As your consciousness settles into the patient rhythms of these liquid-filled vessels, your breathing becoming as steady and measured as the gradual accumulation of morning dew, you begin to appreciate the sophisticated chemistry that pitcher plants have developed to create their aquatic traps. The fluid within each pitcher is not simply collected rainwater, but rather a carefully regulated solution that the plant actively maintains through specialized secretory cells that line the interior walls of the trap. The pH of pitcher plant fluid is typically quite acidic, ranging from 2.5 to 4.5, depending on the species and environmental conditions. This acidity serves multiple functions in the operation of the trap. First, it creates an environment that inhibits the growth of bacteria and fungi that might otherwise decompose trapped prey before the plant can extract nutrients from it. Second, the acidic conditions help maintain the stability of the digestive enzymes that the plant produces to break down captured organisms. Third, the acidity contributes to the preservation of the trap structure itself, preventing the accumulation of organic matter that might clog or damage the pitcher's internal surfaces. The enzyme systems that pitcher plants have evolved to digest their prey represent some of the most sophisticated biochemical machinery in the plant kingdom. These plants produce a cocktail of proteases, lipases, and other digestive enzymes that can break down complex organic molecules into simple compounds that the plant can absorb and utilize. The production and regulation of these enzymes requires precise control mechanisms that ensure the digestive process proceeds efficiently without damaging the plant's own tissues. The surface tension of the liquid in pitcher traps plays a crucial role in their effectiveness. The plant carefully regulates the chemical composition of its pitcher fluid to maintain optimal surface tension characteristics that create a deceptively stable-looking surface, while actually providing very little support for small insects that might attempt to land on it. This creates a situation where insects that approach the liquid surface expecting to find solid footing instead find themselves breaking through into the depths of the trap. The walls of pitcher plants are masterpieces of surface engineering, with different zones that serve distinct functions in the trapping process. The upper portions of the pitcher, near the opening, are often covered with downward-pointing hairs or waxy deposits that make it difficult for insects to maintain their grip and climb back out once they have entered the trap. The middle sections typically have very smooth, slippery surfaces that provide no purchase for climbing legs or claws. The lower portions, where the liquid begins, often have specialized cells that actively secrete digestive enzymes and absorb nutrients from the digestion process. The coloration and patterning of pitcher plants serve important functions in attracting prey to the traps. Many species display bright colors, intricate patterns, or color contrasts that create visual signals that insects interpret as indicators of food sources. Red, purple, and yellow pigments are common in pitcher plants, often arranged in patterns that guide the eye toward the pitcher opening. These visual attractants work in conjunction with chemical attractants to create irresistible invitations that draw insects from considerable distances. The nectar production systems of pitcher plants demonstrate the sophisticated balance these plants maintain between providing genuine benefits to their visitors and creating the conditions necessary for occasional capture. Many pitcher plants produce real nectar around the rim of their pitchers and on specialized structures near the trap opening. This nectar serves as legitimate food for many insects, creating a complex relationship where the plant provides actual nutritional rewards to most of its visitors while capturing only a small percentage of them for its own nutritional needs. The timing of nectar production is often carefully coordinated with local insect activity patterns, with peak nectar secretion occurring during periods when target prey species are most active. This temporal coordination demonstrates the remarkable degree to which pitcher plants have evolved to integrate their biology with the ecology of their local environments, creating hunting strategies that work in harmony with natural behavioral patterns rather than fighting against them. The diversity of pitcher plant species reflects the many different ways that the basic pitcher trap design can be modified and optimized for different environments and prey types. Some species produce small, delicate pitchers that specialize in capturing tiny flying insects, while others create large, robust traps that can handle prey as large as small frogs or lizards. Some species produce their pitchers at ground level, while others hold them high on climbing vines that can reach into the forest canopy. The seasonal development of pitcher plants follows predictable patterns that reflect both the plant's internal biological cycles and the environmental conditions of their habitats. New pitchers typically develop in spring as temperatures warm and insect activity begins to increase. The pitchers gradually mature throughout the growing season, reaching peak effectiveness during the summer months when insect populations are at their highest. As autumn approaches and insect activity declines, many species begin to reduce their investment in pitcher maintenance and prepare for winter dormancy. The internal ecosystems that develop within pitcher plants represent fascinating examples of how complex ecological relationships can emerge within very small spaces. While the primary function of pitcher fluid is to serve as a digestive medium, many pitchers also host communities of bacteria, protozoa, and even small invertebrates that contribute to the breakdown of captured prey. These microbial communities create miniature food webs within the pitchers, with some organisms feeding directly on captured prey, while others feed on the waste products of the digestive process. The mosquito larvae that develop in some pitcher plants represent particularly interesting examples of organisms that have evolved to exploit the resources available within these traps. These specialized larvae can survive in the acidic, enzyme-rich environment of pitcher fluid, feeding on captured prey and organic debris while avoiding being digested themselves. In return, their feeding activities help break down large prey items into smaller particles that the plant can more easily process, creating a mutualistic relationship that benefits both partners. The water balance of pitcher plants requires constant regulation to maintain optimal trap function. Too little water in the pitchers reduces their effectiveness as drowning traps, while too much water could dilute the digestive enzymes and reduce the efficiency of the digestion process. Plants regulate their water levels through a combination of active secretion, selective absorption, and passive evaporation, creating systems that can maintain relatively stable fluid levels despite varying environmental conditions. The root systems of pitcher plants are often quite reduced compared to those of non-carnivorous plants, reflecting their decreased dependence on soil-based nutrition. These simplified root systems are typically sufficient for anchoring the plant and absorbing water, but they contribute relatively little to the plant's overall nutrition. This anatomical simplification represents one of the most visible signs of how carnivory has allowed these plants to redirect their energy and resources away from conventional plant nutrition strategies. The photosynthetic systems of pitcher plants continue to function at high efficiency despite the plant's carnivorous adaptations. The pitcher structures themselves often contribute to photosynthesis, with their green walls containing chlorophyll and actively producing sugars through the conversion of sunlight and carbon dioxide. This dual function, serving as both photosynthetic organs and trapping structures, demonstrates the remarkable efficiency with which these plants have integrated their various biological functions. The pollination systems of pitcher plants are typically quite conventional, despite their unusual feeding habits. Most species produce flowers that are held well above their pitcher traps, ensuring that beneficial pollinators are not accidentally captured. The flowers often display bright colors and produce nectar rewards that attract bees, flies, and other pollinating insects. This separation of reproductive and feeding functions ensures that pitcher plants can maintain healthy populations of the pollinators they need for sexual reproduction. The seed dispersal mechanisms of pitcher plants reflect their adaptation to wetland environments where suitable habitat patches may be widely separated. Many species produce small, light seeds that can be dispersed by wind or water to colonize new bog environments. Some species have evolved specialized seed structures that allow them to float for extended periods, increasing their chances of reaching suitable habitat patches across the landscape. The conservation challenges facing pitcher plants are closely tied to the preservation of their wetland habitats. Many bog ecosystems have been drained, filled, or otherwise modified for human use, leading to significant declines in pitcher plant populations. Climate change poses additional threats, as changing precipitation patterns and temperature regimes can disrupt the delicate water balance that these plants require. The specialized nature of bog ecosystems means that they may be particularly vulnerable to environmental changes, making the conservation of existing habitats crucial for maintaining pitcher plant populations. The horticultural appeal of pitcher plants has led to their cultivation by gardening enthusiasts around the world, but their specialized requirements make them challenging plants to grow outside their natural habitats. Successful cultivation requires careful attention to water quality, humidity levels, soil chemistry, and seasonal temperature variations. The popularity of these plants and cultivation has also created conservation concerns, as wild collection has reduced some natural populations and introduced genetic diversity issues in cultivated lines. The research applications of pitcher plants extend far beyond basic botany and ecology. The enzyme systems these plants have evolved are being studied for potential applications in biotechnology and medicine. The surface properties of pitcher walls are providing inspiration for the development of new materials with novel friction and adhesion characteristics. The fluid dynamics of pitcher traps are contributing to our understanding of how surface tension and viscosity can be manipulated for engineering applications. As you continue to observe these liquid pools of patience, your consciousness now fully integrated with the gentle rhythms of their operation, you can appreciate the profound elegance of their design. Each pitcher represents a perfect balance of chemical, physical, and biological processes working together to create structures that are simultaneously beautiful, functional, and remarkably efficient. The patience these plants demonstrate, waiting days, weeks, or even months for prey to discover their traps, reminds us that some of nature's most effective strategies are also among its most gentle and unassuming. The pitcher plants around you continue their quiet work of maintaining their traps, secreting enzymes, regulating fluid levels, and creating the precise chemical and physical conditions necessary for their unique form of nutrition. Their success demonstrates that in nature, as in life, some of the most powerful strategies are those that work with natural forces rather than against them, creating opportunities through patient preparation rather than aggressive pursuit. As you settle deeper into peaceful observation, your breathing now synchronized with the gentle rhythms of carnivorous plant life, let your attention drift toward perhaps the most famous and fascinating of all carnivorous plants, the Venus flytrap. This remarkable organism has captured human imagination like no other plant, not because it displays violence or aggression, but because it demonstrates something almost magical: the ability of a seemingly passive plant to respond with remarkable speed and precision to the presence of potential prey. The Venus flytrap represents nature's mastery of sensitivity, timing, and gentle but effective action. Picture yourself now kneeling in the sandy, acidic soils of the Carolina coastal plains, where Venus flytraps grow naturally in the wild. The plant before you appears deceptively simple at first glance: a small rosette of leaves growing close to the ground, each leaf ending in what looks like a tiny green mouth fringed with delicate spines. These modified leaves, called traps, rest open like welcoming hands, their bright red interior surfaces glistening with tiny droplets of nectar that catch the morning light like scattered jewels. The beauty of the Venus flytrap lies not in dramatic displays of power, but in the exquisite precision of its trigger mechanism. Each trap contains several tiny sensory hairs, usually 3 to 5 per trap, that serve as the plant's detection system. These trigger hairs are marvels of biological engineering, sensitive enough to detect the gentle footsteps of a small insect, yet sophisticated enough to avoid responding to false alarms like raindrops, fallen debris, or casual contact with other objects. As your consciousness settles into harmony with these remarkable plants, your breathing becoming as measured and precise as their trigger mechanisms, you begin to appreciate the sophisticated logic that governs their operation. The Venus flytrap doesn't simply snap shut at the first touch of its trigger hairs. Instead, it requires multiple stimuli within a specific time window, typically 2 or more hair triggers within about 20 seconds. This built-in delay prevents the plant from wasting energy by closing on objects that aren't actually prey. The mechanism behind the trap closure represents one of the most rapid movements in the plant kingdom, yet it operates not through muscle contractions like animal movements, but through one of the most elegant hydraulic systems in nature. When the proper trigger sequence is detected, specialized cells along the midrib of the trap rapidly lose water pressure, causing the trap lobes to snap shut in a fraction of a second. This hydraulic mechanism allows a plant with no nervous system to respond faster than many animals with sophisticated neural networks. The actual closing motion of a Venus flytrap is breathtakingly gentle despite its speed. The trap lobes curve inward like cupped hands coming together, the marginal spines interlocking to create a temporary prison that holds captured prey while still allowing small, non-beneficial creatures to escape through the gaps. This selective permeability ensures that the plant captures organisms large enough to provide meaningful nutrition while allowing smaller creatures that might not justify the energy cost of digestion to go free. The initial closure of the trap is just the beginning of a remarkably sophisticated process. Once closed, the trap doesn't immediately begin digestion. Instead, it enters a testing phase where it monitors the continued movement of whatever has been captured. If the trapped object continues to struggle and stimulate the trigger hairs from inside the closed trap, the plant interprets this as confirmation that it has captured living prey worthy of digestion. If no further stimulation occurs, the trap will gradually reopen over the course of several hours, releasing whatever non-living material might have triggered the initial closure. This secondary confirmation system demonstrates the remarkable sophistication of the Venus flytrap's decision-making process. Without any brain or nervous system, the plant has evolved the ability to distinguish between living prey that will provide nutritional benefit and non-living objects that would waste digestive resources. This biological logic system operates entirely through chemical and mechanical processes, yet it rivals the decision-making capabilities of much more complex organisms. The digestive process that follows successful prey capture is a marvel of biochemical engineering that unfolds with patient thoroughness over several days. Once the plant has confirmed that it has captured suitable prey, specialized glands within the trap begin secreting a cocktail of digestive enzymes, including proteases that break down proteins, lipases that process fats, and other enzymes that can reduce complex organic molecules to simple compounds that the plant can absorb and utilize. The trap transforms itself into a temporary stomach, creating a sealed digestive chamber where enzymes can work efficiently without being diluted or washed away. The interior surfaces of the trap are lined with absorption cells that can take up the products of digestion, transporting nutrients throughout the plant's body just as the digestive system of an animal transports nutrients through the bloodstream. This process typically takes 5 to 12 days to complete, depending on the size of the prey and environmental conditions. The energy calculations involved in Venus flytrap operation reveal the remarkable efficiency of their hunting strategy. Each trap closure requires a significant expenditure of energy, and the production of digestive enzymes represents an additional metabolic cost. However, the nutritional value of successfully captured prey more than compensates for these expenditures, providing the plant with essential nutrients like nitrogen and phosphorus that are scarce in its native habitat. This positive energy balance is what makes the Venus flytrap's seemingly expensive hunting strategy evolutionarily viable. The seasonal cycle of Venus flytrap activity reflects the plant's integration with its natural environment and the availability of prey throughout the year. New traps develop most actively during the spring and early summer when insect activity is increasing and the plant's nutritional needs are highest. During this period, individual plants may maintain a dozen or more active traps simultaneously, creating multiple opportunities for prey capture as the plant invests heavily in growth and reproduction. As summer progresses and environmental conditions become more stressful, Venus flytraps may reduce the number of active traps they maintain, focusing their energy on supporting their most efficient trapping structures rather than spreading resources across many traps. This strategic resource allocation demonstrates the plant's ability to adjust its hunting effort based on environmental conditions and internal energy balance. The autumn period brings a gradual reduction in trap sensitivity and activity as the plant begins preparing for winter dormancy. Existing traps may remain functional for several more weeks, but new trap development ceases and the plant begins redirecting energy toward root storage and winter survival strategies. This seasonal adjustment ensures that the plant doesn't waste energy maintaining expensive trapping mechanisms during periods when prey availability is declining. The winter dormancy period is crucial for Venus flytrap health and long-term survival. During this time, the plant's above-ground portions may die back completely, while the root system and growing point remain alive but inactive beneath the soil surface. This dormancy period is not simply a time of inactivity, but rather a period of physiological preparation for the next growing season, when the plant will need to rapidly produce new traps and begin hunting again. The microenvironment within Venus flytrap habitats reveals the specific conditions that these remarkable plants require for optimal health and hunting effectiveness. The sandy, acidic soils of their native range provide excellent drainage while maintaining the low nutrient conditions that give carnivorous plants their competitive advantage. The high humidity typical of coastal plain environments ensures that the plants don't suffer from excessive water loss during the hot summer months when trapping activity is at its peak. The fire ecology of Venus flytrap habitats plays an important role in maintaining the open, sunny conditions that these plants require. Periodic wildfires prevent the encroachment of woody vegetation that would shade out the low-growing flytraps, while the nutrient-poor soils that result from repeated burning continue to favor carnivorous plants over conventional vegetation. This fire-maintained ecosystem represents a delicate balance that has persisted for thousands of years. The pollination biology of Venus flytraps demonstrates the plant's need to balance its carnivorous lifestyle with its reproductive requirements. The flowers are produced on tall stalks that hold them well above the trapping leaves, ensuring that beneficial pollinators are not accidentally captured. The flowers themselves are quite conventional, producing nectar rewards and displaying white petals that attract a variety of flying insects including bees, flies, and butterflies. The timing of Venus flytrap flowering is carefully coordinated with local pollinator activity and environmental conditions. Flowers typically appear in late spring or early summer, when pollinator populations are building, but before the hottest part of the summer, when both plants and insects face increasing environmental stress. This timing ensures maximum reproductive success, while avoiding conflicts between the plant's need for pollinators and its carnivorous feeding requirements. The genetic diversity of wild Venus flytrap populations has become a conservation concern as natural habitats are lost to development and other human activities. Venus flytraps have an extremely limited natural range, occurring only within about a 90-mile radius of Wilmington, North Carolina. This restricted distribution makes the species particularly vulnerable to habitat loss and environmental changes that could affect their specialized ecosystem requirements. The horticultural popularity of Venus flytraps has created both opportunities and challenges for conservation. While cultivation has raised awareness of these remarkable plants and provided economic incentives for their preservation, it has also led to overcollection from wild populations and the development of cultivated strains that may lack the genetic diversity of wild plants. Sustainable cultivation practices and habitat preservation efforts are both necessary to ensure the long-term survival of this iconic species. The research applications of Venus flytrap biology extend far beyond botany and plant science. The rapid closure mechanism has inspired engineers working on biomimetic materials and devices that need to respond quickly to environmental stimuli. The trigger hair system has provided insights into plant sensory biology and the evolution of rapid response mechanisms in organisms without nervous systems. The digestive enzymes produced by these plants are being studied for potential applications in biotechnology and medicine. The educational value of Venus flytraps cannot be overstated. These plants provide tangible, observable examples of complex biological processes that help students understand concepts like stimulus response mechanisms, energy balance in biological systems, and the relationship between form and function in living organisms. The rapid, visible response of trap closure makes Venus flytraps ideal subjects for hands-on biological education and scientific observation. The philosophical implications of Venus flytrap behavior challenge traditional distinctions between plants and animals, passive and active organisms, and simple and complex life forms. These plants demonstrate that sophisticated behavior and decision-making capabilities can emerge from purely chemical and physical processes, without requiring nervous systems or brains. This insight has implications for our understanding of intelligence, consciousness, and the nature of life itself. As you continue to observe these remarkable plants, you're breathing now as gentle and measured as their patient waiting between prey captures. You can appreciate the profound lesson they offer about the power of precise timing and appropriate response. The Venus flytrap doesn't waste energy with constant activity or random movements. Instead, it maintains a state of perfect readiness, responding with lightning speed when the proper conditions are met, but remaining perfectly still when action is not warranted. The tender snap of the Venus flytrap represents one of nature's most elegant solutions to the challenge of combining plant and animal characteristics in a single organism. These plants have achieved something remarkable: the ability to hunt actively while remaining rooted in place, to make complex decisions without brains, and to move rapidly while relying entirely on plant physiology. Their success demonstrates that innovation in nature often comes not from abandoning existing strategies, but from finding creative ways to combine and enhance them. The Venus flytraps around you continue their patient vigil, each trap poised and ready, their trigger hairs sensitive to the slightest appropriate touch. They embody a perfect balance of patience and action, stillness and movement, simplicity and sophistication that offers profound lessons about living effectively in harmony with natural rhythms and environmental opportunities. As you drift into even deeper tranquility, your breathing now flowing like morning dew settling on delicate surfaces, let your awareness turn to perhaps the most quietly beautiful of all carnivorous plant strategies, the adhesive traps of Sundews. These remarkable plants have mastered the art of gentle persuasion, creating surfaces so appealing and seemingly harmless that insects are drawn to them like visitors to a welcoming garden party, only to find themselves embraced by the most tender and persistent of holds. Sundews represent nature's understanding that sometimes the softest touch can be the most effective, and that true strength often lies not in dramatic action, but in quiet, patient persistence. Picture yourself now in a sun-dappled bog meadow where sundews grow like living jewels scattered across the moss and peat. Each plant appears to be adorned with hundreds of tiny diamonds, dewdrops that catch and scatter the morning light in rainbow spectrums that seem almost too beautiful to be natural. But these are not ordinary dewdrops. They're the sundew's secret weapon, mucilaginous droplets that serve as both beacon and snare, combining visual beauty with functional effectiveness in one of nature's most elegant hunting strategies. The leaves of sundews are covered with specialized structures called tentacles, not the aggressive arms of sea creatures, but delicate, hair-like projections that wave gently in the slightest breeze, like sea anemones swaying in a tidal pool. Each tentacle is tipped with a gland that produces a drop of clear, slightly sticky fluid that glistens in the light and moves slowly in response to changes in humidity and temperature. These droplets are the sundew's invitation to the insect world, a promise of nectar, a suggestion of moisture, a hint of sweetness that proves irresistible to flying creatures in search of sustenance. As your consciousness settles into harmony with these glistening gardens, your breathing becoming as gentle and persistent as the sundew's quiet embrace, you begin to appreciate the sophisticated chemistry that creates these adhesive traps. The mucilage produced by sundew tentacles is not simple tree sap or plant resin, but rather a complex biological polymer with properties that have been fine-tuned by millions of years of evolution to achieve the perfect balance of stickiness, durability, and digestive capability. The molecular structure of sundew mucilage includes long-chain carbohydrates that create its adhesive properties, proteins that contribute to its elasticity and strength, and enzymes that begin the digestive process almost immediately upon contact with captured prey. This multifunctional fluid serves simultaneously as trap, prison, and digestive chamber, demonstrating remarkable efficiency in biological design. The mucilage maintains its effectiveness across a wide range of environmental conditions, remaining sticky in both humid and dry weather, and retaining its adhesive properties for weeks or months as the tentacles slowly age and are replaced by new growth. The mechanics of sundew capture are breathtakingly gentle, yet absolutely effective. When a small insect lands on a sundew leaf, attracted by the glittering droplets and perhaps expecting to find nectar or water, it instead finds its feet, legs, or wings becoming progressively more entangled in the sticky mucilage. The initial contact might seem minor, just a slight resistance that an insect might notice but not find alarming. However, as the creature attempts to free itself, it comes into contact with additional tentacles, each one contributing more adhesive strands to the growing web of capture. The struggle itself becomes part of the trapping mechanism, as each movement spreads the mucilage further across the insect's body and brings more tentacles into contact with the prey. The sundew doesn't need to move quickly or dramatically to secure its capture. The very efforts of the prey to escape serve to strengthen the bonds that hold it. This self-reinforcing trap demonstrates nature's understanding that sometimes the most effective strategies are those that use the target's own energy against it. The response of sundew tentacles to captured prey reveals another layer of sophistication in these seemingly simple plants. When prey is captured, the tentacles that are not in direct contact with a struggling insect begin to slowly bend toward the capture site, a process that can take anywhere from a few minutes to several hours, depending on the size of the prey and the species of sundew. This movement, called thigmotropism, allows the plant to maximize the number of tentacles in contact with captured prey and ensures that digestive enzymes are delivered efficiently to all parts of the meal. The bending movement of sundew tentacles is one of the most visible examples of plant movement that doesn't rely on rapid changes in cell pressure like the Venus flytrap's snap mechanism. Instead, sundew tentacles achieve their movement through differential growth rates on opposite sides of the tentacle stem, creating a gradual curving motion that appears almost deliberate in its precision. This growth-based movement is much slower than hydraulic movements, but it's also more permanent and doesn't require the immediate energy expenditure that rapid movements demand. The digestive process in sundews unfolds with the same patient thoroughness that characterizes all aspects of their biology. The enzymes contained within the mucilage begin breaking down prey tissues immediately upon capture, working gradually to convert complex proteins and other organic molecules into simple compounds that the plant can absorb through the surface of its tentacles. This external digestion process allows sundews to process prey much larger than they could handle if they had to engulf their food, like Venus flytraps do with their closed trap chambers. The absorption of nutrients from digested prey occurs through specialized cells in the tentacle glands and leaf surfaces that can transport dissolved organic compounds directly into the plant's vascular system. This direct absorption mechanism is remarkably efficient, allowing sundews to extract virtually all available nutrients from their captured prey while leaving behind only the most indigestible components like insect wing cases and other chitinous structures. The diversity of sundew species demonstrates the many ways that the basic adhesive trap strategy can be modified and optimized for different environments and prey types. Some sundews, like the common round-leaved sundew, produce relatively small leaves with short tentacles that specialize in capturing tiny flying insects like gnats and mosquitoes. Others, like the great sundew, can produce leaves several inches long with correspondingly longer tentacles that can handle larger prey, including small flies and even occasional spiders or other arthropods. The tropical sundews represent some of the most spectacular variations on the adhesive trap theme, with some species producing climbing stems that can reach several feet in length, their leaves positioned to intercept flying insects at various heights above the ground. These climbing sundews demonstrate how the basic sundew strategy can be adapted to exploit three-dimensional hunting opportunities in environments where competition for ground-level prey is intense. The seasonal growth patterns of sundews reflect their adaptation to the variable conditions of bog environments and the seasonal availability of insect prey. Most temperate sundews produce their largest and most effective leaves during the late spring and early summer, when insect activity is at its peak. As summer progresses and environmental conditions become more challenging, the plants may produce smaller, more drought-resistant leaves that require less energy to maintain while still providing some hunting capability. The winter survival strategies of sundews vary considerably depending on their geographic location and local climate conditions. Many temperate species form hibernacula, tight, dormant buds that can survive freezing temperatures while the plant's above-ground portions die back completely. These hibernacula contain all the genetic and nutritional resources necessary to produce a complete new plant when favorable conditions return in spring, representing one of nature's most compact and efficient survival packages. The relationship between sundews and their insect prey represents a fascinating example of ecological balance that has evolved over millions of years. While sundews do capture and consume insects, they typically take only a tiny fraction of the total insect population in their habitats. Many insects that visit sundews successfully obtain nectar or other resources without being captured, creating a complex relationship where the plants provide genuine benefits to some visitors while obtaining nutrition from others. The pollination biology of sundews illustrates the delicate balance these plants must maintain between their carnivorous lifestyle and their reproductive needs. Like other carnivorous plants, sundews typically hold their flowers well above their trapping leaves on long stalks that ensure beneficial pollinators are not accidentally captured. The flowers themselves are often quite small and inconspicuous compared to the showy trapping leaves, but they produce adequate nectar rewards to attract the small flies, bees, and other insects needed for successful reproduction. The seed production and dispersal strategies of sundews reflect their specialization for bog and wetland environments. Many species produce tiny seeds that can be dispersed by wind or water across considerable distances, allowing them to colonize new suitable habitats as they become available. The small size of sundew seeds also allows individual plants to produce large numbers of offspring, increasing the chances that some will find suitable growing conditions despite the patchy distribution of appropriate bog habitats. The conservation status of sundews varies considerably by species and geographic region, but many face threats from habitat destruction, water level changes, and competition from invasive species that can alter the nutrient balance of bog ecosystems. The specialized nature of bog environments makes them particularly vulnerable to human activities, and the restoration of damaged bog ecosystems is often difficult and expensive, making habitat preservation the most effective conservation strategy for most sundew species. The horticultural cultivation of sundews has become increasingly popular among carnivorous plant enthusiasts, but their specialized requirements make them challenging subjects for indoor growing. Successful sundew cultivation requires attention to water quality, humidity levels, light intensity, and seasonal temperature variations that mimic their natural bog environments. Many sundew species also require winter dormancy periods that can be difficult to provide in indoor growing situations. The research applications of sundew biology span multiple scientific disciplines, from materials science studies of their adhesive properties to ecological research on predator-prey relationships in bog ecosystems. The mucilage produced by sundews has inspired development of new biomimetic adhesives and medical applications, while their rapid enzyme production systems are being studied for potential biotechnology applications. The biomechanical properties of sundew mucilage represent a particularly active area of research, as scientists seek to understand how these plants achieve adhesive properties that remain effective across such a wide range of environmental conditions. The combination of stickiness, elasticity, and digestive capability in a single biological material represents a level of multifunctional design that current human technology struggles to match. The ecological role of sundews in bog ecosystems extends beyond their direct predation on insects to include their contributions to nutrient cycling and habitat structure. The nutrients that sundews extract from captured prey eventually return to the bog ecosystem through leaf decomposition and root exudates, helping to maintain the nutrient cycling processes that keep these specialized environments healthy and productive. The phenological timing of sundew growth and reproduction is closely coordinated with the seasonal patterns of insect activity and environmental conditions in bog ecosystems. This temporal coordination demonstrates the remarkable degree to which these plants have evolved to integrate their biology with their local environments, maximizing their hunting effectiveness while minimizing conflicts with their reproductive and survival needs. As you continue to observe these glistening gardens of patience, you're breathing now as gentle and persistent as the dewdrops that crown each tentacle. You can appreciate the profound lesson that sundews offer about the power of gentle persistence. These plants don't rely on dramatic movements or aggressive strategies to secure their prey. Instead, they create conditions so appealing and welcoming that prey comes to them willingly, and they maintain their gentle embrace with such patient persistence that escape becomes impossible. The sticky, sweet invitations of sundews remind us that in nature, as in life, the most effective strategies are often those that work with natural tendencies rather than against them. By creating genuine attractions that provide real benefits to many visitors while capturing only what they need for survival, sundews demonstrate a kind of ecological wisdom that combines effectiveness with sustainability, power with gentleness, and success with grace. As you sink deeper into peaceful contemplation, your breathing now flowing like gentle currents in still water. Let your awareness drift beneath the surface of quiet bog pools and pond edges, where one of nature's most ingenious and microscopic hunters operates in complete silence. Here, in the hidden underwater realm that exists just below the mirror-like surface of these tranquil waters, bladderworts have created what might be the most sophisticated trapping mechanisms in the plant kingdom. Microscopic chambers that work like living quicksand, creating powerful suction forces that can capture prey in less than a millisecond while operating on scales so small that entire dramas of capture and digestion unfold in spaces smaller than the period at the end of this sentence. Picture yourself floating just beneath the surface of a quiet bog pond. Your consciousness shrunk to microscopic proportions so that you can witness the remarkable world of these aquatic carnivorous plants. The bladderworts around you appear as delicate, thread-like stems suspended in the water column like living lace, their finely dissected leaves creating intricate patterns that filter sunlight into dancing shadows on the pond bottom. Scattered throughout this underwater garden are thousands of tiny structures that look like miniature urns or bladders, each one no larger than a pinhead, yet containing within it one of the most remarkable biomechanical devices in the natural world. These bladders represent the culmination of evolutionary engineering applied to the challenge of hunting in an aquatic environment where traditional plant trapping strategies simply won't work. Unlike terrestrial carnivorous plants that can rely on gravity, surface tension, or adhesive properties to capture prey, underwater hunters must work within the fluid dynamics of aquatic environments where everything floats, moves constantly, and where traditional physical barriers provide little impediment to escape. As your awareness settles into the gentle rhythms of underwater life, your breathing becoming as quiet and measured as the slow circulation of pond water, you begin to appreciate the remarkable physics that makes bladderwort traps possible. Each bladder operates as a sophisticated suction device that works by creating a pressure differential between its interior and the surrounding water. The bladder walls contain specialized cells that actively pump water out of the interior chamber, creating a partial vacuum that stores mechanical energy like a compressed spring waiting to be released. The door mechanism of bladderwort traps represents one of the most precise examples of biological engineering in nature. Each bladder entrance is sealed by a flexible trapdoor that is held shut by the pressure differential between the low-pressure interior and the normal-pressure water outside. This door is hinged along one edge and held in place by a complex arrangement of molecular scale latches that can withstand the considerable forces created by the pressure differential, while still remaining sensitive enough to open in response to the gentle touch of microscopic prey. The trigger mechanism that opens these underwater traps involves antenna-like structures that extend from around the door opening like tiny whiskers, sampling the water for the presence of suitable prey. When microscopic organisms like water fleas, mosquito larvae, or tiny worms contact these trigger structures, they disturb the delicate balance that holds the door closed. The response is instantaneous and irreversible. The door swings open, water rushes in to equalize the pressure differential, and any organisms in the vicinity are swept into the bladder chamber by the powerful inflow current. The speed of bladderwort trap operation rivals that of Venus flytraps, but operates through entirely different physical principles adapted to the aquatic environment. While Venus flytraps use rapid changes in cell pressure to create their snapping motion, bladderworts rely on stored mechanical energy in the form of reduced pressure within their trap chambers. This stored energy can be released almost instantaneously when the door opens, creating suction forces that can draw in water and prey at speeds approaching 1 meter per second. Remarkable velocity for such tiny structures. The capture process itself is both violent and brief, yet so small in scale that it creates no disturbance in the peaceful underwater environment. The entire event, from trigger activation to door closure to the settling of captured prey within the bladder, takes place in less than a millisecond and affects a volume of water smaller than a grain of sand. From a human perspective, these dramatic capture events are completely invisible, contributing to the serene appearance of bladderwort-inhabited waters. The interior of a bladderwort trap represents a highly specialized digestive chamber that has been optimized for processing the tiny aquatic organisms that make up the plant's diet. The bladder walls are lined with glands that secrete digestive enzymes specifically tailored to break down the proteins, lipids, and other organic compounds found in zooplankton and other microscopic prey. The chemical environment within the bladder is carefully regulated to maintain optimal conditions for enzyme activity while preventing the growth of bacteria or fungi that might interfere with the digestion process. The diversity of bladderwort species reflects the many different ways that the basic suction trap strategy can be adapted to various aquatic environments and prey types. Some species produce tiny bladders specialized for capturing single-celled organisms and the smallest multicellular creatures, while others create larger traps capable of handling prey as large as mosquito larvae or small water fleas. The size and sensitivity of the traps are precisely matched to the size and behavior of the available prey in each species' specific habitat. Terrestrial bladderworts represent a fascinating adaptation of the aquatic suction trap strategy to land-based environments. These species grow in constantly moist soil or among moss, where their tiny bladders can capture soil-dwelling organisms like protozoa, rotifers, and nematodes that live in the thin films of water that coat soil particles. The physics of their trapping mechanism remains essentially the same as their aquatic relatives, but the prey spectrum and environmental challenges are quite different. The seasonal activity patterns of aquatic bladderworts are closely synchronized with the life cycles of their microscopic prey. Many species show peak trapping activity during spring and early summer when zooplankton populations are at their highest levels. The plants invest heavily in trap production during these periods, sometimes maintaining thousands of active bladders simultaneously to maximize their capture efficiency when prey is most abundant. The energy economics of bladderwort trapping reveal the remarkable efficiency of their hunting strategy. While each individual trap is extremely small and captures only tiny prey, the sheer number of traps that a single plant can maintain allows it to harvest significant amounts of nutrition from the microscopic organisms that most plants cannot access at all. A single mature bladderwort plant may maintain several thousand active traps, creating a vast harvesting network that can process enormous numbers of microscopic prey organisms. The role of bladderworts in aquatic food webs extends far beyond their direct predation on zooplankton and other microscopic organisms. By controlling populations of these tiny creatures, bladderworts help regulate the complex ecological relationships that maintain the health and stability of aquatic ecosystems. Their selective pressure on zooplankton communities can influence algae populations, nutrient cycling, and even the behavior and distribution of larger aquatic organisms. The water quality requirements of bladderworts reflect their adaptation to the specific chemical conditions found in bog ponds and similar acidic, nutrient-poor aquatic environments. These plants thrive in soft water with low mineral content and pH levels that would be challenging for most aquatic plants. Their ability to supplement their nutrition through carnivory allows them to prosper in waters that lack the dissolved nutrients that conventional aquatic plants require. The photosynthetic systems of bladderworts continue to function efficiently despite their specialization for carnivory. The finely divided underwater leaves provide substantial surface area for light capture and gas exchange, while their thin construction minimizes the energy investment required for their maintenance. This efficient photosynthetic system provides the energy needed to operate the numerous suction traps while supporting the plant's growth and reproduction. The reproductive strategies of bladderworts often involve both sexual and asexual reproduction methods that reflect their adaptation to aquatic environments. Many species can reproduce vegetatively through fragmentation, allowing pieces of the plant that break off to develop into new individuals. This asexual reproduction provides a reliable way to maintain populations in stable habitats, while sexual reproduction through flowers and seeds allows for genetic diversity and long-distance dispersal to new habitats. The flowering of aquatic bladderworts provides some of the most delicate and beautiful displays in bog ecosystems. The flowers are typically held above the water surface on slender stalks, creating ethereal displays of yellow, purple, or white blooms that seem to float on air above the water. These flowers are perfectly conventional in their structure and function, producing nectar rewards for pollinators and developing seeds through normal sexual reproduction processes. The winter survival strategies of temperate bladderworts involve various forms of dormancy and energy conservation that allow them to survive freezing temperatures and reduced food availability. Some species form specialized overwintering buds called turions that sink to the bottom of ponds where they remain dormant until spring warming triggers their return to active growth. These turions represent remarkably compact survival packages that contain all the genetic and nutritional resources needed to regenerate complete plants. The ecological indicators that bladderworts provide make them valuable species for monitoring the health of aquatic ecosystems. Their presence typically indicates clean, acidic water conditions with healthy populations of microscopic organisms, while their absence may suggest pollution, eutrophication, or other environmental problems that have disrupted the delicate balance of bog pond ecosystems. The conservation challenges facing bladderworts are closely tied to the preservation of their specialized aquatic habitats. Many bog ponds and similar acidic wetlands have been drained, filled, or degraded by human activities, leading to declines in bladderwort populations throughout their ranges. The restoration of degraded wetlands can sometimes allow bladderwort populations to recover, but the specific water chemistry and biological conditions they require make restoration efforts particularly challenging. The biomimetic applications of bladderwort trap mechanisms have inspired engineers working on microfluidic devices and other technologies that require precise control of fluid movement at microscopic scales. The combination of stored mechanical energy, rapid activation, and selective operation that characterizes bladderwort traps represents design principles that could be applied to various technological applications, from drug delivery systems to environmental monitoring devices. The study of bladderwort ecology has contributed significantly to our understanding of predator-prey relationships in aquatic ecosystems and the role of carnivorous plants in nutrient cycling. These microscopic hunters process enormous numbers of tiny organisms that would otherwise be inaccessible to larger predators, creating important links in aquatic food webs that help maintain ecosystem stability and productivity. The sensory biology of bladderworts reveals remarkable sensitivity and discrimination capabilities that allow these plants to distinguish between suitable prey and non-beneficial objects. The trigger mechanisms can detect the gentle movements of living organisms while ignoring debris, air bubbles, and other objects that might accidentally contact the trap structures. This discrimination ability helps ensure that the plants don't waste energy by activating their traps unnecessarily. The chemical ecology of bladderwort habitats includes complex interactions between the plants and the microbial communities that develop within and around their traps. Some bacteria and other microorganisms appear to contribute to the digestive processes within the bladders, while others may compete with the plants for nutrients or prey. These microbial relationships add another layer of complexity to bladderwort ecology and demonstrate the interconnected nature of bog pond ecosystems. The physiological adaptations that allow bladderworts to function as aquatic carnivorous plants include specialized systems for gas exchange, mineral absorption, and waste elimination that work efficiently in underwater environments. These adaptations required significant modifications to basic plant physiology and represent some of the most sophisticated examples of plant adaptation to aquatic carnivory. As you continue to float peacefully in this microscopic underwater realm, your consciousness now attuned to the gentle rhythms of aquatic life, you can appreciate the profound elegance of bladderwort hunting strategies. These plants have solved the challenge of carnivory in an aquatic environment through mechanisms so sophisticated and efficient that they rival the engineering capabilities of human technology, yet they operate with the quiet grace and environmental harmony that characterizes all successful natural systems. The living quicksand of bladderwort traps reminds us that nature's most powerful mechanisms often operate on scales and time frames that make them invisible to casual observation. The drama of predation and the complexity of ecosystem relationships continue unabated in realms so small that millions of individual events can occur within a single drop of water. each one contributing to the larger patterns of energy flow and nutrient cycling that maintain the health and beauty of aquatic ecosystems. As you continue to drift in perfect tranquility, you're breathing now as gentle and steady as the gradual changes that mark the passage of seasons in nature's most patient gardens. Let your awareness turn to one of the most subtle and understated of all carnivorous plant strategies, the quiet, greasy embrace of butterworts. These remarkable plants have perfected what might be called the art of passive persistence, creating leaf surfaces so perfectly engineered for gentle retention that insects find themselves held not by dramatic traps or violent mechanisms, but by something as simple and inescapable as the slow, steady grip of a caring hand that never lets go. Picture yourself now in a mountain meadow or limestone cliff face where butterworts grow like living green platters scattered across the moss and rocks. Each plant appears deceptively simple, a flat rosette of thick, succulent leaves that seem to glow with an inner light, their surfaces appearing slightly wet or oily, as if they have been lightly brushed with the finest coating of natural wax. The leaves lie flat against the ground or rock surface, creating what looks like a peaceful landing pad for any small creature that might choose to rest upon their welcoming surfaces. The beauty of butterworts lies in their complete lack of drama or obvious predatory features. Unlike the snap traps of Venus flytraps or the glistening adhesive droplets of Sundews, butterworts present themselves to the world as nothing more threatening than slightly glossy leaves that might provide a convenient resting place for tired insects. This understated approach represents one of nature's most elegant examples of camouflage functionality, where the most effective trap is the one that doesn't look like a trap at all. The leaf surface of a butterwort is a masterpiece of biological engineering that operates through principles so subtle they remain mysterious to scientists for many years. The upper surface of each leaf is covered with thousands of tiny glands that produce a thin film of oily, mucilaginous secretion. This secretion is not sticky in the obvious way that sundew mucilage is sticky, but rather creates a surface with unique physical properties that make it almost impossible for small insects to gain the traction they need to walk or fly away once they have landed. As your consciousness settles into harmony with these masters of subtle retention, your breathing becoming as calm and measured as their patient embrace, you begin to understand the sophisticated physics that makes butterwort traps so effective. The oily secretion creates what material scientists would recognize as a low-friction surface that provides almost no grip for tiny insect legs and feet. When a small fly or gnat lands on a butterwort leaf, expecting to find a normal plant surface that will support its weight and provide traction for movement, it instead finds itself on a surface as slippery as ice, unable to gain purchase for takeoff or even for walking to the edge of the leaf. The chemical composition of butterwort secretions reveals a complex mixture of compounds that work together to create the perfect combination of properties needed for effective prey retention. The oily components reduce surface friction and prevent insects from gaining traction, while mucilaginous compounds provide just enough adhesion to prevent prey from being blown away by wind, while still allowing the plant to maintain its innocent, non-threatening appearance. The glandular system that produces butterwort secretions operates continuously throughout the growing season, maintaining a fresh, effective surface that can function in a wide range of weather conditions. Unlike adhesive traps that might lose their effectiveness when washed by rain or dried by sun, butterwort surfaces are constantly renewed and refreshed, ensuring consistent trapping performance regardless of environmental conditions. The digestive process in butterworts unfolds with the same patient thoroughness that characterizes every aspect of their biology. Once prey is securely held on the leaf surface, specialized glands begin secreting digestive enzymes that work gradually to break down the captured organisms. This process can take several days or weeks to complete, proceeding so slowly and gently that captured insects often remain visibly intact for extended periods while their internal tissues are gradually processed and absorbed. The absorption of nutrients from digested prey occurs through the same glandular system that produces the trapping secretions, creating an elegant closed-loop system where the same structures that capture prey also process and absorb the resulting nutrients. This integration of trapping and digestive functions demonstrates remarkable efficiency in biological design, minimizing the energy investment required to maintain separate systems for capture and nutrition extraction. As you sink even deeper into peaceful observation, your awareness now flowing like gentle streams that slowly reshape landscapes through patient persistence, let your consciousness turn to one of nature's most remarkable demonstrations of transformation, the process by which carnivorous plants convert captured prey into the nutrients they need for growth and survival. This process of digestion represents not destruction or violence, but rather one of the most elegant examples of recycling and renewal in the natural world, where complex organic molecules are gently broken down and rebuilt into new forms of life. The digestive systems that carnivorous plants have evolved represent some of the most sophisticated biochemical engineering in the plant kingdom, rivaling in complexity the digestive systems of many animals while operating through entirely different mechanisms adapted to plant physiology and lifestyle. These systems must accomplish the challenging task of breaking down complex animal tissues while protecting the plant's own cells from damage, all while operating in environments that range from acidic bog pools to alkaline mountain soils. Picture yourself now observing this transformation process as it unfolds within the various trapping structures we have explored. In the liquid depths of pitcher plants, enzymes work like gentle molecular scissors, carefully snipping the bonds that hold proteins together and releasing amino acids that the plant can absorb and use to build its own proteins. The process resembles the slow dissolution of sugar in warm tea, with complex structures gradually becoming simpler until they can pass through cellular membranes and enter the plant's transport systems. The enzyme systems that drive carnivorous plant digestion are marvels of molecular specificity and efficiency. Each enzyme is precisely designed to break particular types of chemical bonds while leaving others intact, allowing the plant to extract useful nutrients while avoiding the breakdown of its own cellular structures. Proteases cut protein chains at specific points, lipases process fats and oils, and carbohydrates handle complex sugars and starches, each working in perfect coordination with the others to ensure complete and efficient digestion. The pH regulation that accompanies carnivorous plant digestion demonstrates the remarkable precision with which these plants control their internal chemistry. Most digestive processes require specific pH levels to function effectively, and carnivorous plants have evolved sophisticated mechanisms for creating and maintaining these optimal chemical conditions within their trapping structures. The acidic environment of pitcher plant fluid, for example, is carefully maintained through the secretion of organic acids and the selective absorption of basic compounds. The protective mechanisms that prevent carnivorous plants from digesting themselves represent another marvel of biological engineering. The same enzymes that can break down captured prey could potentially damage the plant's own tissues if they were not carefully controlled and contained. Plants accomplish this protection through a combination of enzyme inhibitors, protective surface coatings, and careful spatial separation of digestive and living tissues. The timescales involved in carnivorous plant digestion reflect the patient, thorough approach that characterizes all aspects of their biology. While animal digestive systems might process a meal in hours, plant digestive systems typically require days or weeks to completely break down captured prey. This extended processing time allows for maximum nutrient extraction and ensures that virtually nothing useful is wasted in the conversion process. The nutrient absorption mechanisms that operate during digestion involve specialized transport systems that can move dissolved organic compound from the site of digestion to other parts of the plant where they're needed for growth and metabolism. These transport systems must be able to handle a wide variety of molecular types and sizes, from simple amino acids to complex organic compounds, all while maintaining the selectivity needed to exclude potentially harmful substances. The integration of digestion with the plant's overall metabolism demonstrates the remarkable coordination that characterizes carnivorous plant physiology. The nutrients obtained from prey digestion must be incorporated into the plant's existing metabolic pathways, converted into appropriate forms for storage or immediate use, and distributed throughout the plant's tissues according to current needs and priorities. As you continue to observe these processes of gentle transformation, you're breathing now as patient and thorough as the digestive processes themselves. You can appreciate the profound wisdom that carnivorous plants demonstrate in their approach to nutrition and resource utilization. Rather than wasteful or destructive, their predation represents one of nature's most elegant examples of efficient recycling, where nothing is truly lost, but only transformed from one form of life to another. The seasonal regulation of digestive activity in carnivorous plants shows how these processes are integrated with the plant's overall life cycle and environmental conditions. During periods of active growth, digestive systems operate at peak efficiency to support the plant's nutritional needs, while during dormancy periods, digestive activity may slow or cease entirely as the plant conserves energy for survival rather than growth. The evolutionary origins of carnivorous plant digestion reveal how these sophisticated systems developed gradually from simpler beginning points, with each step in the evolutionary process providing immediate benefits that justified the energy investment required for further development. The pathway from simple enzyme secretion to complex digestive systems demonstrates how evolutionary processes can create remarkable complexity through the accumulation of small, beneficial changes over vast periods of time. The ecological role of carnivorous plant digestion extends beyond the individual plants to influence entire ecosystem nutrient cycles. By capturing nutrients from mobile prey and eventually returning those nutrients to the ecosystem through leaf decomposition and root exudates, carnivorous plants serve as important links in the complex networks of nutrient flow that maintain ecosystem health and productivity. The conservation implications of understanding carnivorous plant digestion highlight the importance of maintaining the complex ecological relationships that these plants require for survival. Changes in prey populations, water chemistry, or environmental conditions can all affect the efficiency of digestive processes and ultimately influence the health and survival of carnivorous plant populations. As you rest in peaceful contemplation of these remarkable transformation processes, you can appreciate how carnivorous plants have solved one of biology's fundamental challenges: how to obtain essential nutrients in environments where they are scarce, through mechanisms that are both elegant and effective, demonstrating that some of nature's most powerful solutions are also among its most gentle and patient. As you drift into the deepest layers of tranquil awareness, your breathing now flowing with the ancient rhythms that govern all life on Earth, let your consciousness expand to encompass the grand temporal patterns that shape the lives of carnivorous plants throughout the turning of seasons. These remarkable organisms do not exist in a static state of perpetual hunting, but rather follow intricate cycles of activity and rest, abundance and scarcity, growth and dormancy that mirror the larger patterns of seasonal change and demonstrate nature's profound wisdom in balancing energy expenditure with resource availability. Picture yourself now observing a bog garden through the course of an entire year, watching as the subtle but profound changes of each season transform both the plants and their hunting strategies. Spring arrives with a gradual warming that awakens dormant carnivorous plants from their winter rest, triggering the slow unfurling of new leaves and the careful construction of fresh trapping structures. This awakening happens not with sudden urgency, but with the measured pace of organisms that have learned to synchronize their biological rhythms with the reliable patterns of seasonal change. The spring emergence of carnivorous plants represents one of nature's most elegant examples of energy management and resource allocation. After months of dormancy, these plants must carefully balance their energy expenditures, investing in new growth while maintaining enough reserves to survive potential late-season cold snaps or resource shortages. The first new leaves to emerge are often smaller and simpler than those produced later in the season, reflecting the plant's cautious approach to returning to active growth and hunting. As warming temperatures trigger increased insect activity, carnivorous plants begin the gradual process of constructing and activating their hunting apparatus. Venus flytraps develop new traps with fresh trigger hairs and fully charged hydraulic systems. Pitcher plants begin secreting the enzyme-rich fluids that fill their chambers. Sundews produce their first tentacles crowned with glistening droplets of adhesive mucilage. Each species follows its own precise timing, evolved over millions of years to synchronize trap readiness with prey availability. The chemistry of spring activation in carnivorous plants involves complex cascades of hormonal signals and metabolic changes that coordinate the transition from dormancy to active growth. Plant hormones like auxins and gibberellins regulate cell division and elongation, while specialized enzymes begin producing the digestive compounds that will be needed for prey processing. This biochemical awakening must be precisely timed to avoid wasting energy on hunting structures before prey becomes available. As your awareness settles into the patient rhythms of seasonal change, your breathing becoming as regular and predictable as the annual cycles that govern carnivorous plant life, you begin to appreciate how these plants have learned to work with rather than against the natural patterns of resource availability in their environments. Spring represents not just an awakening, but a careful preparation for the intensive hunting season that lies ahead. The early summer period brings the peak of carnivorous plant activity, as warming temperatures and lengthening days create optimal conditions for both plant growth and insect reproduction. This is the season when trapping structures reach their maximum effectiveness, when digestive systems operate at peak efficiency, and when the energy investment in carnivory pays its highest dividends in captured nutrients. The bog gardens shimmer with the maximum number of active traps, each one perfectly maintained and ready to take advantage of abundant prey. The synchronization between carnivorous plant activity and insect life cycles represents one of evolution's most sophisticated examples of temporal coordination. Many bog insects have evolved life cycles that peak during the same periods when carnivorous plants are most active, creating a complex ecological dance where predator and prey populations rise and fall in coordinated patterns that maintain ecosystem balance while providing optimal feeding opportunities for the plants. The midsummer period often brings challenges that test the resilience and adaptability of carnivorous plant hunting strategies. High temperatures and potential drought conditions can stress both plants and prey, requiring adjustments in trap maintenance and hunting behavior. Some carnivorous plants respond by producing smaller, more drought-resistant traps, while others may temporarily reduce their hunting activity to conserve energy for basic survival needs. The late summer transition marks the beginning of a gradual shift in carnivorous plant priorities, as the focus begins to change from maximum nutrient acquisition to preparation for the challenges of autumn and winter. New trap production may slow as plants redirect energy toward root storage and the development of overwintering structures. Existing traps continue to function, but their maintenance may become less intensive as plants begin conserving resources for the dormant season ahead. The autumn period brings a complex series of physiological changes that prepare carnivorous plants for winter survival. Deciduous species begin the process of nutrient translocation, moving valuable compounds from leaves that will be shed to roots and other storage organs that will survive the winter. The production of new trapping structures typically ceases, while existing traps may remain functional for several more weeks, taking advantage of late-season insect activity. The timing of autumn preparation varies considerably among different carnivorous plant species and reflects their specific evolutionary adaptations to local climate patterns. Some species begin preparing for winter as early as late summer, while others continue hunting activities well into autumn. This variation in timing demonstrates the fine-tuned nature of seasonal adaptations and the importance of precise coordination with local environmental conditions. The formation of overwintering structures represents one of the most remarkable aspects of carnivorous plant seasonal cycles. Many species produce specialized dormant buds, underground stems, or other structures that can survive freezing temperatures while maintaining the genetic and nutritional resources needed to regenerate the entire plant in spring. These overwintering structures are marvels of biological engineering, containing everything necessary for survival and renewal in incredibly compact packages. The winter dormancy period is not simply a time of inactivity, but rather a carefully regulated state of reduced metabolism that allows carnivorous plants to survive adverse conditions while preparing for the next growing season. During dormancy, cellular repair processes continue at reduced rates, energy reserves are carefully managed, and physiological systems are maintained in states of readiness for rapid activation when favorable conditions return. The regional variations in carnivorous plant seasonal cycles reflect the diverse climate conditions under which these plants have evolved. Tropical species may experience less dramatic seasonal changes, but still show responses to wet and dry seasons or other environmental cycles. Arctic and alpine species may have extremely compressed growing seasons that require rapid activation and intensive activity during brief favorable periods. The metabolic adjustments that accompany seasonal changes in carnivorous plants involve complex regulatory mechanisms that control enzyme production, hormone levels, and cellular activity. These adjustments must be precisely coordinated to ensure that energy is allocated appropriately between hunting activities, growth, reproduction, and survival needs as conditions change throughout the year. The reproductive timing of carnivorous plants is closely integrated with their seasonal hunting cycles, with most species producing flowers during periods when their nutritional status is optimal and when environmental conditions favor successful pollination and seed development. This coordination ensures that reproduction occurs when plants have adequate resources to support the energy-intensive processes of flower and seed production. The seed dormancy and germination strategies of carnivorous plants reflect their adaptation to seasonal environments where timing is critical for survival. Many species produce seeds with built-in dormancy periods that prevent germination until environmental conditions are suitable for successful establishment. These dormancy mechanisms help ensure that young plants emerge at times when they can take advantage of optimal growing conditions. The phenological studies of carnivorous plants have revealed subtle but important changes in seasonal timing that may be related to climate change and other environmental factors. Some species are showing earlier spring emergence or delayed autumn dormancy, changes that could affect their synchronization with prey species and their overall fitness in changing environments. The conservation implications of carnivorous plant seasonal cycles highlight the importance of protecting entire ecosystems rather than just individual plant populations. The complex relationships between carnivorous plants, their prey, and their seasonal environments require habitat preservation efforts that maintain natural hydrological cycles, temperature patterns, and ecological relationships. The horticultural challenges of replicating natural seasonal cycles for carnivorous plants in cultivation demonstrate the complexity and importance of these temporal patterns. Successful cultivation often requires careful attention to temperature cycling, photoperiod changes, and dormancy requirements that mirror the natural seasonal patterns these plants have evolved to follow. The research applications of studying carnivorous plant seasonal cycles extend beyond plant biology to include climate science, ecology, and conservation biology. These plants serve as sensitive indicators of environmental change and provide valuable data for understanding how organisms adapt to seasonal variation and climate fluctuations. The philosophical implications of carnivorous plant seasonal cycles offer profound lessons about patience, timing, and the value of working in harmony with natural rhythms rather than fighting against them. These plants demonstrate that success in nature often comes not from constant activity, but from knowing when to be active and when to rest, when to invest energy in growth, and when to conserve resources for survival. As you continue to observe these seasonal patterns, you're breathing now as steady and reliable as the annual cycles that govern carnivorous plant life. You can appreciate the deep wisdom embedded in their approach to temporal resource management. They teach us that there is a time for hunting and a time for rest, a time for growth and a time for conservation. And that true effectiveness often comes from perfect synchronization with the natural rhythms that govern all life on Earth. The seasonal cycles of carnivorous plants remind us that hunger itself can be seasonal, that the need for supplemental nutrition varies with environmental conditions and internal physiological states. These plants have learned to intensify their hunting efforts when conditions are optimal and to reduce them when resources are scarce or when other priorities take precedence. This flexible approach to carnivory demonstrates the sophisticated integration of feeding behavior with overall life strategy that characterizes successful organisms in seasonal environments. The bog gardens around you continue their eternal cycle of seasonal change, with each turning of the year bringing new opportunities for growth and challenges for survival. The carnivorous plants that call these places home have mastered the art of living in harmony with these cycles, demonstrating that some of nature's most effective strategies are those that embrace change rather than resist it, that work with natural patterns rather than against them, and that understand the profound importance of timing in all aspects of life. As you settle into the deepest peace of this gentle journey, you're breathing now as soft and natural as morning mist rising from still water. Let your consciousness drift into the timeless tranquility of the bog at twilight. Here, as the last light of day fades into the gentle embrace of night, the carnivorous plants around you enter their own quiet period of rest, their hunting activities slowing to the patient rhythms that have sustained these remarkable organisms for millions upon millions of years. In this peaceful transition from day to night, you can sense the profound harmony that connects all life in these ancient, sacred spaces. Picture yourself now sitting quietly beside a bog pool as evening settles over the landscape like a soft blanket of contentment. The water reflects the deepening sky with perfect clarity, doubling the beauty of the twilight and creating a sense of infinite depth and possibility. The carnivorous plants around you, pitcher plants, sundews, Venus flytraps, and all their patient relatives, continue their gentle work of maintaining their traps and processing their nutrients, but with the measured pace of organisms that understand the value of working in harmony with natural rhythms rather than fighting against them. The evening chorus of the bog begins as daylight fades. The soft calls of night birds, the gentle splash of small creatures moving through the water, the almost inaudible whisper of evening breezes stirring the delicate leaves and trapped structures of the carnivorous plants. This symphony of gentle sounds creates a natural lullaby that speaks of the deep peace and contentment that characterizes these special places where predation has evolved into something remarkably like hospitality, where hunting has become an art of patient invitation rather than aggressive pursuit. As your awareness settles completely into this tranquil evening scene, your breathing, now perfectly synchronized with the subtle rhythms of bog life, you begin to understand the profound lessons that carnivorous plants offer about finding success through gentleness, achieving power through patience, and creating abundance through working in harmony with natural limitations rather than fighting against them. These remarkable plants have discovered that some of nature's most effective strategies are also among its most graceful and sustainable. The conservation message embedded in the story of carnivorous plants speaks to the importance of protecting not just individual species, but entire ecosystems and the complex relationships that sustain them. These plants can thrive only in environments where water chemistry, soil conditions, seasonal patterns, and prey populations all exist in delicate balance. Their preservation requires protecting entire watersheds, maintaining natural fire cycles, and preventing the pollution and habitat destruction that can disrupt these ancient, finely tuned ecological relationships. The scientific insights gained from studying carnivorous plants continue to inspire new technologies and deepen our understanding of biological processes. From the biomimetic materials inspired by pitcher plant surfaces to the medical applications of plant-derived enzymes, these remarkable organisms contribute to human knowledge and welfare while reminding us of the incredible creativity and ingenuity that evolution has produced through millions of years of patient experimentation. The philosophical teachings of carnivorous plants extend far beyond biology to offer profound insights into effective living. They demonstrate that true strength often lies in stillness rather than constant activity, that the most powerful strategies may be the most gentle, and that success often comes to those who can wait patiently for the right opportunities rather than those who pursue their goals with aggressive haste. The relationship between carnivorous plants and their prey exemplifies the kind of ecological balance that characterizes healthy natural systems. While these plants do capture and consume other organisms, they do so in ways that support rather than threaten the overall health of their ecosystems. They take only what they need, provide genuine benefits to many of their visitors, and contribute to the nutrient cycling and habitat complexity that support diverse communities of organisms. The beauty of carnivorous plants lies not just in their unusual feeding habits, but in their demonstration of how life can find creative solutions to challenging problems through patient adaptation rather than force. In environments where conventional plant nutrition strategies fail, these remarkable organisms have evolved hunting techniques that are both effective and sustainable, both powerful and gentle, both sophisticated and elegantly simple. As you rest in the gathering darkness of the bog evening, your consciousness now completely at peace with the gentle rhythms of these ancient places, you can feel yourself becoming part of the larger patterns of natural harmony that carnivorous plants embody. Your breathing matches the slow circulation of bog water. Your heartbeat synchronizes with the patient pulse of seasonal cycles, and your thoughts settle like dew forming on the delicate surfaces of sundew tentacles. The dreams that inhabit these bog gardens are dreams of perfect adaptation, of finding abundance in apparent scarcity, of achieving remarkable success through strategies based on invitation rather than aggression. These are dreams of ecological wisdom that has been refined over geological ages, dreams of sustainability that could persist for millions more years if we have the wisdom to protect the habitats where they flourish. The carnivorous plants around you continue their quiet work as night deepens, their traps poised and ready, their digestive processes proceeding with patient thoroughness, their metabolic systems preparing for another day of gentle hunting and harmony with the natural world. They embody a kind of success that requires no boasting, no display of dominance, no disruption of the peaceful environment they call home. Let your own dreams now merge with the dreams of the bog, carrying with you the lessons of patience and persistence that carnivorous plants teach. Rest in the knowledge that true effectiveness often comes through gentle persistence rather than dramatic action, that the most powerful transformations may be the most gradual, and that some of nature's most remarkable achievements arise from working with natural forces rather than fighting against them. Sleep peacefully in the embrace of this ancient wisdom, knowing that the bog gardens will continue their eternal cycles of growth and rest, hunting and digestion, abundance and conservation that have sustained these remarkable ecosystems for countless generations. The carnivorous plants will continue their patient vigil, their gentle invitations, their quiet transformations that demonstrate how life can flourish even in the most challenging environments through creativity, patience, and profound harmony with the natural world. Dream of sticky dewdrops that sparkle like jewels in morning sunlight, of pitcher plants that welcome visitors with gracious hospitality, of Venus flytraps that respond with perfect timing to the gentle touch of trigger hairs. Dream of the microscopic suction chambers of bladderworts, of the quiet embrace of butterwort leaves, of all the countless gentle mechanisms that carnivorous plants have evolved to find abundance in environments where other forms of life struggle merely to survive. The bog dreams with you now, its ancient wisdom flowing through your consciousness like water flowing through peat and moss, carrying with it the accumulated knowledge of millions of years of successful adaptation to challenging environments. Rest in this wisdom, knowing that you carry within you the same capacity for creative problem-solving, patient persistence, and gentle effectiveness that allows carnivorous plants to thrive in their specialized niches. The gentle hunters of the bog continue their eternal dance of invitation and transformation, reminding us that some of life's most profound lessons come not from the dramatic and obvious, but from the subtle and patient, not from the forceful and aggressive, but from the gentle and persistent. Sleep well, knowing that tomorrow will bring new opportunities to apply these lessons of natural wisdom to the challenges and opportunities of daily life. Always remembering that true success often comes to those who, like carnivorous plants, have learned the art of gentle persistence and harmony with the natural rhythms that govern all life on Earth.